DNA and Cell Function, Section 4. Last time we looked at the codon chart. Let's start by reviewing today the overall process of protein synthesis so it doesn't get lost. What you see here is a simplified diagram showing protein synthesis. So this whole process is known as protein synthesis. And it begins with DNA. Where is DNA found? Well, if it's a eukaryotic cell, it's found in the nucleus. The first step of 2 in protein synthesis is when the DNA unwinds one section known as a gene, and it makes, using one side of that gene, an mRNA transcript. And this step, because it's making an mRNA transcript, is known as transcription. It makes that transcript by base pairing. Don't forget, there is no T in RNA. One way to remember that is don't run. So if you've got the R in A, there is no T. It's going to use a U instead. The RNA then leaves the nucleus and moves to the cytoplasm, which you can see shown here. This is the same RNA that was made in transcription. In the cytoplasm, it's going to meet up with a ribosome, and that ribosome is going to allow it to put together amino acids like this methionine, abbreviated MET. They will be bonded to each other in a chain by peptide bonds. And when they are all done and the chain of amino acids is complete, it will release and fold to form a protein, which is sometimes called a polypeptide because of the many peptide bonds. So it has taken the transcript and translated it into a protein. And that is why this second stage is called translation. And this diagram is an overview, very simplified, of protein synthesis. This is how it should happen. But what happens when a mistake is made? A mistake in DNA is called a DNA or gene mutation. And DNA mutations have a big impact on protein synthesis. Remember that DNA is the code the cell uses to put the right amino acids in the right sequence. So if the code is bad, then the protein can be bad. We call that an abnormal protein. Or if the code is bad, the cell might not be able to use it. It might not be able to make any protein at all. So gene mutations, DNA mutations, can lead to abnormal, which means they're not going to function like they should, proteins, or no proteins where one should be made. Well, what kind of mutations happen? There are 2 basic groups of mutations that you need to know. The first is the point or substitution mutation. It happens at just one point, one base in the DNA. And the second is the frameshift mutation. This is where a base gets added in insertion or a base gets deleted in deletion, and it changes the length of the DNA. Point mutations and frameshift mutations can both lead to abnormal or no proteins. But frameshift mutations, especially if they happen early in the gene, cause a lot of change, whereas point mutations will change just one amino acid. That can still mean the protein is abnormal or doesn't get made. But point mutations happen at just one point, whereas frame shifts change the gene from that place where the mutation happened all the way to the end of the gene. So what are the two basic types of DNA mutation? They are the point mutation, also called the substitution, where one base is changed, and the frame shift, where a base is added or deleted from the sequence, which changes the length of the gene. Let's take a closer look. Gene mutations happen because the DNA has a mistake, and we'll study later how that happens. For now, I just want to focus on what it means for protein synthesis. If the DNA mutation happens in a cell that is a body cell, what we call a somatic cell, like a lung cell, a liver cell, something like that, it can be bad, and we'll see exactly what it can do in a few minutes. But the good news is it will not be passed on to offspring because we don't use liver cells or heart cells or brain cells to make babies. The cells we use to make babies are special cells called gametes. If the gene mutation happens in the gamete cell, the one used to make a baby, it can be passed on to the offspring. So when you think about mutations, it's important to think about what cell are they happening in, as well as what are they going to do to that cell. How is a somatic cell different from a gamete? Well, you can see right away, somatic cells have all of the chromosomes in that organism's genome, and gametes just have half. That's because a gamete cell will fuse with another cell to make the baby. So a somatic cell is a body cell like heart, liver, lung, and has all of the chromosomes of the genome. Gametes have half. What causes DNA mutations? Well, mutagens or mutagenic agents are things that can cause the DNA to mutate. What are some mutagens? Radiation, chemicals, and infectious agents like certain viruses or bacteria can all cause DNA mutations. For example, UV radiation from the sun can cause DNA mistakes in the DNA of skin cells where the UV radiation penetrates. It can cause thymines that are side by side to link up together in what we call a thymine-thymine dimer. And then when the DNA is used, it can't be used correctly because the thymines are stuck together. X-rays, which are really relatively safe, but too much can also lead to the same type of DNA mutation. Chemicals, in general, we're surrounded by chemicals, they make up everything, are not always mutagens, but there are some chemicals that have been found to cause DNA gene mutations. Some of those chemicals that we know cause gene mutations can be found in cigarette smoke, nitrate or nitrite preservatives used in your food. They can be found when some chemicals are heated to high temperatures, like in barbecuing, or in products that we use, like benzoyl peroxide in acne medicine. And infectious agents, based on the way that they work, if they are interfering with the DNA of the host cell, which is the cell they're infecting, can also cause havoc and create gene mutations in the DNA of the host cell. So what are the three types shown? We have radiation, chemicals, and infectious agents. We talked about point versus frame shift. Now let's take a little bit of a closer look at those. Here we have at the top insertion and deletion. These two are frame shift mutations. So what you see here is this DNA is supposed to have 4 bases in this section. In an insertion, an extra base, the A, gets added in. Remember that DNA is used 3 bases at a time. So originally, it was supposed to be using A, C, T. But when the A gets shoved in there, now when the transcript gets made, it's going to have this extra base, which is going to shift everything down. Same thing with the deletion. If instead of adding, you remove a base, everything gets shifted up. Substitution works a little differently. This is a point mutation. In a substitution, one of the original bases gets switched out for a different base. What was supposed to be a T here gets switched out with a G. Let's answer the questions. Which base is added in the insertion? It's the A, the second one. Which base was removed in the deletion? It was the T. Which base got changed in this substitution cartoon? T was changed to a G. What is an insertion? An extra base is added to the gene. What about a deletion? A base is taken away from the gene. Why are substitutions point mutations? They only affect one base, one point of the gene. Well, that is a general overview of mutations. Your work will help you see how that causes change in the function. But if you want to review or have any questions, there's a slide here with lots of resources, including an interactive tutorial, another interactive tutorial, a Quizlet for the vocabulary, and I know there was a lot, and an overview of mutations by the amazing Amoeba sisters. Their video is here. I hope this was helpful. I'll see you next time. Have a great day.